Hello, and welcome to everyone joining us for today's Select Science webinar titled Cruise Through Genomics Quantification in QC with Lunatic. My name is Mathilde Marx. I'll be moderating today's presentation, and I'm delighted to be joined by our guest speaker, Dr. Ross Walton, Senior Application Scientist at Unchained Labs. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of Auditorium and click to open the Q&A window. So, without further delay, I'm delighted to hand over to Dr. Walton. I'd like to thank him for presenting to us today.
Thank you for the introduction, and hello to everyone who's joining us today. I'm looking forward to discussing how you can cruise through genomics, quant, and QC with Lunatic. Recently, I've gotten into a new hobby, mixing cocktails. It's a fun and delicious pastime, but I've found that there are some drinks that just have way too many steps and ingredients. Like this Bloody Mary, for example. If you're curious, it's called the Bloody Beast from Sobelman's in Milwaukee, Wisconsin. I've never tried it, but I really don't think I want to. Fluorescence-based assays for DNA or RNA quantification also have a lot of steps. They require dilutions, they take time, they use costly reagents, and they can introduce pipetting errors, which negatively impact results. Not to mention the occasional intervention for assay optimization. Even for the most routine assays, it's important to find faster, cheaper, and easier methods. Think about how rewarding it would be to figure out a way to get your experiments done under budget and before the deadlines with a simpler, more efficient workflow. Another issue to watch out for is that fluorescent dyes are only linear for a fairly small range of concentrations. At higher concentrations, fluorescent dyes saturate, which makes the process even more complicated. If the samples lie outside of this fluorescent linear range, they need to be diluted. But if you don't know the starting concentration, it can be hard to know exactly how much dilution to use. And this adds more complexity, more steps, and more opportunities for error. Using Lunatic would let you know right away if your samples are good to go. In the experiment shown here, you can see the linearity for samples read using Lunatic across a wide concentration range. And in the Pico green range, the assays agree with each other quite closely. That is, at least until Pico green starts to saturate. Because of the limited dynamic range of a fluorescent method, a sample might have to be diluted multiple times so that it falls into the standard curve. On the other hand, Lunatic offers a wide dynamic range with reproducibility and accuracy without the hassle of standard curves and dilutions. Lunatic is a full-spectrum UV-Vis spectrophotometer, which only requires 2 microliters per sample and no dilution steps, reagents, nor standard curves. Lunatic can measure up to 96 samples at a time in about 5 minutes, and our plates have a standard SBS footprint and a standard well layout, so they're compatible with liquid handling robots and multi-channel pipetters. Lunatic gets results in a single step. Unlike fluorescent dye quantification, just read the samples and then move on to the next important experiments with your quantifications in hand. Lunatic plates make all of its amazing capabilities possible. They have the same well spacing as a 96 well plate, but instead of just a well, each one is connected to a microfluidic circuit. Each microfluidic circuit either has 1 or 2 fixed path length microcuvettes, which cover a wide dynamic range from 0.03 to 275 OD. These circuits ensure that there's no cross-contamination or evaporation. Fixed path lengths give a high level of precision and accuracy to absorbance measurements, which means you know exactly what you're measuring. While you need just a single step to measure a sample with Lunatic, you just load it and go, the instrument itself actually takes a few steps. After you pipette your sample into the well, it's pulled into the reservoir by capillary action. At this point, the sample can sit for up to 2 hours before reading with no evaporation. That makes life a lot easier, especially if you're running lots of samples in a day. Then you load the plate into the instrument, which reads the empty microcuvettes to eliminate any absorbance contribution from the plastic, and a pump applies pressure, which moves the sample into the microcuvettes for reading. Here, we can see exactly what sample loading looks like with the help of some fluorescein and a black light. In 2 wells, we've already added 2 microliters of fluorescein. As you can see, to add another sample, You simply add 2 microliters into that input well, and the sample wicks into the plate. Here's what it looks like before and after a read happens. You can see the fluorescent in the microfluidic reservoirs on the left, and on the right, you can see samples that have already been pulled into the microcuvettes. Now, you might notice that one of those cuvettes is slightly brighter than the other. That's because one of them has a longer path length than the other, which means there's slightly more volume in that sample of the sample in that one, so it's more visible. This combination of 2 different path lengths is part of what gets Lunatic its phenomenal dynamic range. Lunatic easily meets the needs for labs working at larger scales, for example, biobanks or core facilities. For these high throughput needs, the plates used for Lunatic are fully compatible with liquid handling robots and plate handling robots. Lunatic features full robotic integration options based on Scylla or an API, and it has plate tracking with barcodes, ensuring traceability in certified labs as well as sample tracking. Let's take a hypothetical example of a high throughput use case, a biobank that aims to enroll and genotype 50,000 individuals. They compare 2 methods for quantifying the DNA in their workflow, Lunatic or a fluorescent-based assay. The question is, which will be better? If we compare the costs to complete their study using these 2 methods, Lunatic offers significantly lower consumables and labor costs. Consumable cost savings is about $65K in US dollars, and the labor savings is about $78K. The biggest driver of that savings is that Lunatic doesn't need standard curves or any reagents to quantify nucleic acids. That's saved both on consumables and time, since there's no need to dilute out DNA standards or dilute down the samples. If you're curious which method is more accurate, we can actually take a look at that too. You can see the outputs here from one of our biobank customers, where they compared the results of exactly the comparison I'm describing. They quantified thousands of genomic DNA samples with either Lunatic or picogreen, then normalized those DNA samples to 25 nanograms per microliter, and then measured again. You can see that there's much wider variation in the results with picogreen compared to the samples read on Lunatic. And this is due in part to the dilution that was required to read the initial concentration of the sample, introducing error into the workflow. Lunatic doesn't need dilution, so it has a lot less pipetting involved, hence fewer opportunities for error. Lunatic brings in 2% accuracy and 1% precision for its absorbance measurements, which we've validated in several different ways using independently verified standards. First, we have these 3 NIST reference materials that we can use to verify Lunatic's absorbance results: a monoclonal antibody, a tryptophan standard, and a uracil standard. In every case, Lunatic measurements of the standards were within 2% of the NIST certified value, and the CVs of the measurements were within 1%. If you don't know what CV means, it's the same as relative standard deviation, or RSD. You can read about these results and how we did this validation in an app note on our website called Spot On UEVIS Accuracy with Lunatic and Stunner. NIST also offers another standard reference material called SRM2372A, which is a human DNA quantitation standard mainly for the purposes of validating qPCR. It consists of 3 components, each of which has a certified mass concentration and a manufacturer's 95% confidence interval. As you can see, Lunatic's measured concentrations were well within this confidence interval, and it also had CVs of 1% or less. When you look at Lunatic's linearity, accuracy, and precision over a range of concentrations, they're equally impressive. This shows a DNA dilution series from 25 nanograms per microliter up to 1,400 nanograms per microliter. The close agreement between the target concentration and the average measured concentration shows off Lunatic's spot-on accuracy within 2% of the expected value. The low CV values across the dilution range also help show off its precision, typically below 1%. Delivering this dynamic range, linearity, accuracy, and precision is the advantage of using fixed path lengths. Lunatic handles high concentration samples without dilution, with high accuracy and reproducibility. The UV spectra shown here have the telltale absorbance peak at 260 nanometers, which is indicative for purified nucleic acids. The highest concentration of DNA in the dilution series on the left was 12,800 nanograms per microliter, while the highest concentration of the RNA dilution series on the right was 6,800 nanograms per microliter. That's a bit higher than the concentrations you typically expect to see for genomic samples, but it's in the range of possibility for therapeutic molecules. High lunatic plates have a dynamic range from 0.03 OD to 275 OD and are suited for extremely high concentrations, while standard regular lunatic plates max out at 40 OD, which is equivalent to about 2,000 nanograms per microliter of double-stranded DNA. Either one of these two plate types can run on any lunatic without any additional software or any modifications, and you get to pick which one will best suit your needs. If you need something that's very high, measuring things that are very high concentration, a high lunatic plate's more appropriate. But if you're measuring things of a more moderate concentration, it might be that the lunatic plates are more appropriate. Since DNA and RNA samples can be pretty messy, I want to show you how Lunatic's unmix applications let you see annoying impurities that other UV systems might actually mistake as either DNA or as RNA. Here, we're looking at results from a sample of purified DNA in TE buffer that are pretty close to ideal. This is the typical result you'd see for a clean sample. We can see the expected peak maximum at 260 nanometers, and the quality ratios are within the accepted range. For pure DNA, we'd expect an A260 over A230 ratio greater than 2 and an A260 over A280 ratio of about 1.8. Note the accuracy and the reproducibility. We're getting nice low standard deviations and percent CVs for these triplicate reads. Sadly, the real world is often far from our ideal. For example, one of our customers had an issue where they were getting higher DNA concentration measurements by absorbance compared to what they were getting when they used a fluorescent dye. To troubleshoot this, they ran lambda DNA through their purification system and compared the sample using Lunatic before and after running it through that system. Concentrations before and after by unmix were pretty similar, but the sample had significantly higher turbidity after purification, shown by that gray line in the graph on the right. The most likely source of this turbidity was bead carryover from the purification. And bead carryover can interfere with PCR, it can interfere with library ligation, and can actually cause sequencing failures of genomic DNA. The Unmix app successfully deconvoluted the turbidity spectrum to give DNA concentrations that were closer to those that came from the fluorescent dye, but it was faster, cheaper, and didn't require any additional reagents or dilution steps. There's another advantage of Unmix here, which is that it helped us figure out the cause of the unusual signal. That can help you determine if it's a one-off problem with a specific sample, or if it's a process issue that might need to be more closely addressed. Let's look at another example of an impurity that can have serious detrimental impacts on downstream processes and how you can use Unmix in order to detect that impurity and figure out what to do about it. For a second example, we have DNA that's in TE buffer that has way too much EDTA. 5.5 millimolar instead of the more typical 0.5 or 1 millimolar. Now, this is something that's probably more common to have happen at a core facility where people might not know exactly how to prepare their elution buffers, but of course mistakes can happen anywhere. This much EDTA would inhibit the polymerases necessary for PCR and library prep, and ultimately could lead to DNA sequencing failures if it's left unchecked. Fortunately, it's easy to deal with excess EDTA. You just simply repurify the sample and elute the sample again in the proper buffer. In this case, since we have decent DNA yield, repurification is probably worthwhile. However, if we didn't have good yield, you'd probably be better off just throwing the sample away and starting over again. Unmixed makes it easy to determine both the real DNA yield and figure out the source of any kind of unusual signal, enabling informed decision-making about those precious DNA samples and helping you troubleshoot your process. Unmixed can also deconvolute UV absorbances from protein and nucleic acids, or even DNA from RNA, still giving you those real nucleic acid concentrations in that kind of messy mixture of all kinds of molecules. By using Unmixed, we can see exactly what the problem is and what it needs to be solved. Checking preps on Lunatic lets you know exactly what's in your sample, saving you time by throwing up a red flag before you move a sample into downstream applications. Researchers from all over the world are using Lunatic as part of their genomics workflows. Maybe some of these case studies that we're going to go over will actually give you some ideas of how you might be able to use Lunatic in your own workflows. 4 such examples from around the world are that the UK Biobank uses it as part of their large-scale genotyping efforts. The All of Us research program and the Mayo Clinic Biobank are using it as part of their whole genome sequencing workflow as they attempt to recruit at least 1 million participants into a health research resource. Personalis Inc. is using it as part of their validation efforts for Next Personal, which is an ultra-sensitive personalized assay for detecting circulating tumor DNA. And then Aarhus University in Denmark, I hope I'm pronouncing that right, I actually don't know, use it as part of their RNA sequencing core facility workflows. All 4 of these projects, to some degree, use automated Lunatic for quantification and for QC of nucleic acids for genomics research. The UK Biobank is an open resource with genotypic and phenotypic data on approximately 500,000 individuals, and they use Lunatic as part of their genotyping process. This process consists of collecting blood and then extracting DNA using magnetic bead extraction. Then that DNA was aliquoted across 3 different tubes, and the primary aliquot was quantified by Lunatic. If that primary aliquot met the process criteria, then they would genotype that DNA and then store 2 of the aliquots. Those criteria was a DNA concentration of above 10 nanograms per microliter and an A260 over A280 ratio of 1.8 to 2.2. Genotyping results showed that the DNA was of very high quality. This study was just the start. The NHS is currently using samples from the UK Biobank, plus others, as part of their 100,000 Genomes project to streamline whole genome sequencing-based diagnoses of genetic diseases and to discover underlying disease-causing variants. In 2022, they published a paper in Nature with results from pilot studies of this project that gave more than 1,000 patients a genetic diagnosis for their condition, as well as discovered 4 novel disease-causing variants. As part of the UK Biobank study, they were also able to show a strong correlation in readouts of nucleic acid concentration comparing Lunatic to picogreen. And most importantly, what they found was that there was no difference in the quality of the downstream data depending on which measurements you relied on. However, as we've discussed, Lunatic is significantly cheaper and faster than picogreen, as well as provides you that purity information. This result was published in this paper, Welsh et al. In 2017, if you'd like to read about it for yourself. The All of Us research program, which is sponsored by the NIH, uses the Mayo Clinic Biobank and has the goal of enrolling at least 1 million people into a database with health-related information and genetic data. Their process involved automated DNA purification, again from blood, using either salt precipitation or magnetic bead purification. Regardless of which one of these 2 purification methods were used, the DNA was quantified by Lunatic, and if it passed the whole genome sequencing criteria, it was normalized to 50 nanograms per microliter and stored, as well as went through that WGS step. Those passing criteria was a concentration of DNA greater than or equal to 50 nanograms per microliter and an A260 over A280 ratio between 1.6 and 2. The All of Us research program is continuing its mission, and the database that it's generated is actually already being used for all kinds of medical research: type 2 diabetes, COVID-19, stroke risk, pain management, and a lot more. This ongoing project had to receive an IDE from the FDA, and they published a paper in 2022 detailing the application process, as well as information about how they did their sequencing and the data analysis, and all of this information about how they used Lunatic as part of their process. Next Personal is a liquid biopsy assay from Personalis that uses probes made from DNA taken from a patient's tumor as an ultra-sensitive, personalized ctDNA assay for detecting residual disease, treatment efficacy, and recurrence in circulating free DNA, or cfDNA. Lunatic was used to quantify their pre-enrichment library concentration, which was part of their performance specifications that they had during the analytical validation of their assay. And this performance specification was related to the DNA input amount. The result from the analytical validation was that they were able to achieve 100% specificity in detecting ctDNA in the cfDNA of 123 patients. And this assay, this Next Personal assay, is actually currently being used and available for patients and researchers for detecting minimal residual disease, or MRD. And they published this entire analytical validation account in OncoTarget in 2024. Lunatic is also part of a ton of course facilities at universities around the world, and this is just one example. At our host university, there is a sequencing core facility for RNA and for DNA, as well as for, I believe, methylomics and a few other things, called the MoMA, M-O-M-A core facility. They do sequencing studies for the university as well as people outside of the university. In this particular paper, the researchers were examining the transcriptome for tissue-specific effects of sex chromosome aneuploidy, which can give rise to a whole host of different traits and diseases. And Lunatic was a key component of the RNA-seq workflow. After isolating RNA with silica membranes, they eluted that RNA, quantified it by Lunatic, and based off that Lunatic quantification, used 500 nanograms of RNA as the input for their RNA-seq libraries. These stories really do a great job, I think, of highlighting how genomic research is making the dream of personalized medicine a reality, as well as shows off how the wide variety of apps in the Lunatic software really opens up the door to all types of nucleic acid samples. With fluorescence-based quantifications, you would need to get specific reagents and design individualized assays for each one of those studies and each one of those different types of molecules. However, Lunatic is a one-step method for measuring genomic DNA, cell-free DNA, and RNA with a dilution-free, standard-free, and reagent-free workflow. If there's one thing I want to leave you with from today's webinar, it's this. Lunatic is the ultimate genomics quant tool. It's simpler, faster, and easier than other methods without the need for dyes, reagents, or dilutions. If you have any questions, please feel free to ask them today, and I'll be happy to answer them either at the end of the presentation or by reaching out to you directly. Also, remember one additional thing. Lunatic gets you same time, concentration, and quality measurements. With all that time and money you can save with Lunatic, you can finally take the vacation you've always wanted with a nice, simple drink in your hand. Thanks for your time, and I'll be happy to take any questions now.
Thank you, Dr. Walton, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We'll now move on to the last part of today's webinar, the Q&A session. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of auditorium and click to open the Q&A window. So, to kick off this final portion of today's event, So our first question is, what are the dynamic range of Lunatic?
So the dynamic range of Lunatic actually depends on what kind of plate you're using. So for a regular Lunatic plate, the dynamic range is 0.03 OD up to 40 OD. For a high Lunatic plate, it's 0.03 OD to 275 OD. That doesn't quite, I know it's hard to translate probably into, the nucleic acid concentrations, which is, I suspect, what people are actually looking for. And that's because every molecule that you measure on a UV-vis spectrophotometer absorbs at a slightly different amount of light. So taking those absorbance measurements, those OD measurements, translating that into a molecular range or mass concentration range depends on what kind of molecule you're looking at. So for DNA, those high lunatic plates, that 0.03 to 275 OD, equates to a dynamic range of 1.5 up to 13,750 nanograms per microliter. And then for RNA, it's 1.2 to 1,000 nanograms per microliter. That has to do with the differences in how much light DNA and RNA absorb. Double-stranded molecules tend to absorb slightly less light than single-stranded nucleic acids, so double-stranded DNA has a slightly higher dynamic range.
Lovely, thank you. And how long does it take to measure a full plate on the Lunatic?
So with the regular lunatic plates, each one of those circuits that we looked at earlier in a lunatic plate has only a single microcuvette, which makes it read faster, brings that upper concentration down, but that's usually more than high enough for most genomic applications. So for those plates, those lunatic plates, measurement time is about 5 minutes. For high lunatic plates, it's about 10 minutes because you've got two microcuvettes, which of course increases the amount of measurement time.
Great, thank you. And what other kinds of samples can we quantify with the Lunatic?
So the sort of rule of thumb I have with Lunatic is that if it absorbs in the range between 230 and 750 nanometers and is aqueous, you can measure it on Lunatic. So that can be, we've got people who've done genomic DNA and RNA, like I showed, plasmids, cell-free DNA, proteins, antibodies, ADCs, oligos, peptides, and you can even do colorimetric assays since it does have visible light range measurements as well. And we've even had people who've actually quantified certain small molecules depending on what sort of solvents they're dissolved in. So yeah, if it's aqueous and absorbs between 230 and 750 nanometers, you can definitely quantify it on Lunatic.
Lovely. And what impurities can Nmix detect?
That is actually going to vary depending on which unmixed application you use. We actually customize each one of those unmixed applications to detect the most common impurities that are present in those kinds of samples. So, for example, the DNA apps can detect RNA, protein, EDTA, thiocyanate salts, phenols, azides, and some detergents. Now, that's for the DNA applications. It wouldn't really make sense if in an RNA application you're also trying to detect RNA as an impurity. So it's going to depend on what kind of molecule it is, and that affects which of the applications you choose.
And next question says, do I need to blank with a Lunatic, and what should I use?
So, yes, blanks are pretty important when you're measuring UV-vis absorbance. It's an important piece of making sure that you're not measuring absorbance when the absorbance is actually caused by the buffer and not caused by your molecule of interest. So, one of the nice things about Lunatic, though, is that you actually don't have to measure a blank every single time. If you're measuring a batch of samples all in the same buffer, you can just measure that buffer absorbance once as a blank and actually save it and then reuse it. So what kind of blank you use depends on which of the applications you're using. If you're using the unmixed applications, we recommend using water as a blank. But if you're using some of the more standard nucleic acid quantification assays or some of the standard A260 or A280 assays, then we recommend blanking with that buffer. That's particularly true if the buffer has some amount of color to it or something else that might be absorbing in that UV-vis range. You need to be able to correct that out in order to get the most accurate and most precise quantification of your molecule of interest.
Thank you. I think we still have time for another one. So, how
Mhm.
pure do my samples need to be?
So this is actually something that I really love about doing nucleic acid quantification using UV-Vis measurements, and particularly on Lunatic, is that you can actually use that UV-Vis data, that Lunatic data, as part of your QC step for figuring out. So the answer is the sample doesn't necessarily have to be pure because you're actually checking how pure it is as part of the application, as part of the assay. So unmixed makes that really easy. With unmixed, you get concentration measurements while you're figuring out if there are any impurities present and even how much of those impurities are present. And with a little bit of intuition, a little bit of practice, you can actually get a pretty good idea if a given impurity is likely to interfere with any sort of downstream process you might be doing. Is that if that's PCR based or if that's next-gen sequencing based. Then, once you know if a given impurity is going to interfere with that downstream process, you can make an informed decision about how you're going to proceed with a given sample. If the nucleic acid yield is high enough, it might be worth trying to repurify it. If that yield is really high, and then also the amount of impurities is really high, then that sample probably just isn't worth dealing with anymore, and you're better off moving on to the next thing.
Lovely. Thank you so much, Dr. Walton. I'm afraid that is all we have time for today. But I'd like to thank Dr. Walton for today's discussion and presentation and for this lovely Q&A session. And thank you to everyone joining us online for submitting your questions. We hope that you found this a worthwhile session. If we didn't quite get to your question today, we'll follow up with answers to these after the webinar. And if you have any other question after today's session, please feel free to email myself at editor@selectscience.net. If you are interested in receiving a certificate of attendance, this will be shared with everyone who has attended live today via email a few days after the event. And for those watching on demand, a certificate can be requested by emailing us at the same email address. And if you'd like to listen again to today's webinar or invite a friend to do so, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.